and that building soil health can happen really fast, but it doesn't happen directly overnight. It doesn't happen in year one, but we can move in the right direction. So I've got some of the parameters that we'll deep dive into, show some of the data from the group and some of the results from this past year. Then I want to share with you guys what I've been really working on since we were last together, which is the carbon intensity scores. There's some huge things happening, guys, in regards to how do we get paid for doing this stuff, and how do we get paid for telling our story, for the data that we are collecting. There's some tax credits and stuff that we're watching that is a huge, huge deal. So I want to get in that and lead that into where's this stuff going, okay? So let's talk on the Haney test, talk on some of that stuff, but I also want to share with you guys what Continuum has really been working on over the last year and a half in regards to carbon intensity, where I think you guys really need to be thinking about how do you monetize these things that you're doing on your farm as we go into 2024 and beyond. Okay. Further intro on what the heck is Continuum Ag and all that. We're a software company that I started in 2015, and I started it to be able to manage all this Haney soil health test. The Haney data, being able to map it out, being able to take the Haney result and turn it into a variable rate precision application. So that's where we started things, but then it's led into managing our holistic farm data, because the soil data is super important. Measuring soil health is super important, but also the rest of our agricultural system, the rest of the story has some real value to it. So I want to hit on that of how we've continued to expand things. It's this data. As the consumer wants to know about sustainability, as companies want to understand their carbon footprint, we are part of their carbon footprint. We are part of their supply chain, and they're going to be asking us for data, and there's money to be had and getting paid for that data. But it all boils back to having data, utilizing these resources, and making smart decisions on the farm. We do continue to really expand our team and stuff too since I was last with you guys. Been ramping up like crazy. This whole soil health space, hopefully you guys are feeling it. Soil health, sustainability, this whole movement really continuing to pick up speed. So really applaud what you guys have done here with the farmer peer groups and the advocates here. All right. So we got to start, of course, with the soil health principles. Hopefully you guys are hearing these, are understanding how to implement these principles, but we got to start here. And the key thing that I always hit on when we're talking about the soil health principles is these tools have got to be offensive management tools for your farm. Okay, just like you put on that nitrogen fertilizer because it's supposed to return you 1 pound of nitrogen to 1 bushel of corn, right? As we're taught, it's an offensive management tool to get the outcome. These systems also have to be offensive management tools to directly drive a financial outcome. Okay, I believe these systems have been preached a little too much as defensive management tools to defend against water quality problems, defend against erosion, defend against regulation. And you guys know it more than anybody in your neck of the woods here. That's really important to defend against those things. But I think as we communicate to our peers and to our neighbors, we've got to show them not just here's what we're defending, but here's the positive offensive outcomes that we can garner. That cover crop or that reduced tillage system, that's part of the fertilizer management program. It's part of the nutrient stabilization program. It's part of the herbicide program. It's part of the moisture management program, part of the temperature management program, plantability, and now in your insurance program, and now it's part of your carbon program and your ability to get paid based on data, okay? So that's a key thing to start with as you think about these practices on your farm, as we talk about data, make sure that you are thinking about, okay, if I'm going to do this practice, that's great, but how am I directly going to impact my P&L? Because we're running businesses here. We have to make these things pay. We have to make them pay consistently in order to be able to continue to invest in our family farms and get more folks on board doing these practices as well. It's that offense, not defense. It's minding context as we implement these principles to each of our own locations. But that offense piece, I think, is really, really critical. One of the ways that we can go on the offense here is managing that cover crop as part of the fertilizer program. Okay, I think that's really important. That cover crop is going to hold on to nutrients for you. Some cover crops are going to sequester nutrients for you out of the atmosphere, like our legumes that can pull nitrogen out of the atmosphere. Our grasses that can hold on to leftover nutrients and be able to release them back in season the next growing year. The Haney test is something I really like using because it helps me to quantify the nutrients that are actually available to the crop. That's the difference in the Haney test versus a regular traditional Malick analysis that a lot of you guys have done for years. The Haney test shows you your current assets. The current assets, what's readily available right now to the plant. And the reason that it shows you those current assets is because of the extraction that it uses. It uses water, and it uses the H3A extraction, which is it mimics the plant root exudates and what the plant is actually able to interact with. Okay, the inverse of that is like the Malick extraction, Olsen, Bray, some of these traditional analysis. They're very strong acids that essentially show you your intermediate assets or what could come available in the growing season. And we've utilized these analysis because they've been documented with university trials to make fertilizer recommendations based on the analysis. The difference here in the Haney test is it shows you what's actually available right now. What's actually available to the plant in the organic and in the inorganic form. And now you know, here's what's in my soil. We understand how much the crop needs through university recommendations and how many pounds of NPK and other nutrients that it actually takes to produce a bushel and produce the yields that we want. But the Haney test shows you what is actually available in your soils. So that's why as we think about using the Haney test, I recommend doing it in the spring, in season, right around planting time, right around side dress time, as we're making decisions and trying to understand what does that plant actually need. We need to know what is actually available to that plant. And then as you go through like the experiments and some of the data that you'll see here, you can cross compare different scenarios. How much nutrients is actually available where I had that diverse cover crop versus where I didn't have a cover crop? How much nutrients are actually available where I had manure versus where I'm in a synthetic fertilizer program? Those kind of variables. Being able to quantify what is actually available to the plant. A couple other things here as you look through this. So if you guys that were in the program, the soil sampling, we're sending the samples down to Brookside because they're here in Ohio. I also work very closely with the Regen Ag Lab. They're out in Nebraska. But the Haney test from Brookside, I really like their display here. I like their the display and the view of the data. So a couple things on where this data is at for you guys that brought some of the results with you, but just high level of how to read this, and we'll dig into the individual parameters. When I look at the test, what I look at is up in the, I don't know if I've got the pointer's going to work or not, but up in the upper left, it says IR gas analyzer. That's infrared gas analyzer. That number is biological activity. The higher that number, the more biological activity we've got going on in the soil. Microbes breathe. I said they don't have lungs. They respire off, breathe off CO2, just like we do. The longer we sit in this room, we're building up CO2 concentration in this room. And there's a tester called an infrared gas analyzer that we can be holding here in the room. And the longer we're in here, the concentration of CO2 will go up. If we had more people in the room, the rate would go up faster. Also, if we all went running around and were really active and breathing a lot, the rate would go up faster. You follow? Same thing is how we test the microbes in the soil. We take some of the soil at the lab, they dry it down, they put it in a little jar, they put some water in it to mimic a rain. They cap off the jar for 24 hours and they see how much CO2 is released in that jar over 24 hours. The more CO2 that's released, the more microbes that there were in that soil, the more active those microbes. Same type of thing as us sitting in the room here today. Okay, so that's what that first number means. We'll go into some data, but the infrared gas analyzer, microbial activity, the higher that number, the more microbes there are, the more active they are. With the Haney test, that's all it shows you for the microbial piece. It's just how many are there. Other tests like the PLFA analysis or DNA sequencing, some of these other tests, they show you the details of what actual microbes do you have? What specific species, what specific function do they do? The Haney test, it just shows you how active do you got. The activity is driven by the food. How do we get more microbes in the system? We got to feed them. It's the only way we're going to get more. So how do we feed them? What do microbes eat? They eat liquid carbon. The Haney test measures that as water extractable organic carbon, WEOC. Water extractable organic carbon is the organic sugar content that those plant roots are exuding down into the soil. Plants take in CO2 from the air, combine that with sunlight energy to make simple sugars, and they can pump up to 70% of those simple sugars back through the roots to feed the microbes. And we can measure it right here. So as we get more cover crops out there, as we get more perennial systems out there, more diversity out there, the more photosynthesis we have going on, the more carbon we're pumping down into the soil, the more food we have for the microbes, which we measure as WEOC, and therefore the more active the microbes will be. Okay, so WEOC is the food, but we can't just feed them sugar. We can't just feed them carbon. We've got to also give them the nitrogen because they have to be in balance. They have to have a balanced food, balanced diet, just like we do. Water extractable organic nitrogen is essentially that readily available nitrogen molecule that those microbes can use as they produce enzymes and proteins to be able to break down and eat that carbon. In my story, I shared that we lost yield in our first year doing this cover crop. And the reason was we put out all this cereal rye because we were getting paid for it, right? There was a government cost share program. We took the money, said, cool, cereal rye, let's do it. Let's throw it out there. Well, we put on all this carbon into the system and had all this sugar going out there, all this carbon content going out there, but we didn't have any organic nitrogen. We've been in a conventional or synthetic fertilizer system. We were just a corn soybean rotation. Our organic nitrogen content was really low, and we just put all this carbon out there, and because it was not in balance, the microbes couldn't eat all that carbon correctly. And therefore, they took nitrogen away from my corn crop. They took the fertilizer that we put out there intended to feed the corn because they needed nitrogen to break down all of that carbon. But we don't want to have too much nitrogen, or else the microbes have too much energy there, too much nitrogen. They're going to eat all that carbon and then start going to more stable carbon, aka the organic matter. So we have to feed them in balance. And actually this one, a 5.9 is actually a little bit on the higher organic nitrogen side of things, but depends on kind of time of year and things like that. These numbers will fluctuate throughout the year. But point being here, guys, is it's keeping these things in balance. And as we are building soil health, we got to get more food. We got to have a balanced diet out there for the microbes to then improve biological activity over time. So because it's the ratio and the balance, those ranges of optimum and high and low, that kind of levels itself out, that it's looking at the balance and the relationship between the two nutrients rather than the absolute of what's ideal versus less than ideal, which would depend on your CEC, right? So because it's the ratio, that CEC isn't as impactful because it's just looking at the balance and the relationship between the two different nutrients. But again, like for some of these, as you guys look at what your CEC is and the actual nutrient availability, that's where, as you interpret those availability real numbers, that's where you got to factor in the context of your CEC, which this doesn't necessarily do that. I don't know if everyone here knows Mark, but Mark Dodd, he and I have been running in the circles together for a long time here, but we were both together up in Michigan, and then you had hit up guys down here, and it's like, well, let's go to this event here too. But Mark does a ton of these as well. So you want that biological activity to be in relationship with the amount of food that's going in there. It is one of the parameters in the back end, just on this report, they don't necessarily have that. Can go into a couple of the other detailed pieces here. Okay, so when we were looking at here, so now this goes into some of the detailed of the individual components, okay? What we found is that out here in this trial that we did, and the testing that you guys did, we found the average biological activity, the average CO2 burst was 88.9. The best one that we had out here was 286. So, and the worst one was 21.6. So as you guys look at your results, you can kind of compare your results versus the group. These are on the, these slides should be available somewhere. Stephanie, you can share these ones too. But these we showed in the webinars and stuff that we've done as well, if you guys want to go back and see some of these. But on average, 88.9. The best one was 286, if I remember right. It was like a long-term pasture with rotational grazing, like all the soil health principles. You know, it makes sense. Where you have these soil health principles, where you're building up that biological activity, you'll see it reflected here in the CO2 burst. Typically, I mean, it depends on the crop, of course, for this organic nitrogen. But usually I like to see it in that 20, 30, 40 part per million type of range. We take that times 2, that means we got 40 to 80 pounds of nitrogen readily available in the organic form. So the more we can get that readily available organic nitrogen out there, the less we have to supplement. So that's where we've been able to do it at scale. But I like the project you guys had here too of let's just test some trials. Let's look at in the field versus in the fence row, like a native area, just being able to get a little indicator of what's going on. But okay, so instead of doing a double crop, like in you guys' neck of the woods and stuff, of wheat and then beans, a relay crop is we're planting our beans into the cover when it's small. Instead of terminating it and doing delayed termination, we come out there with the combine. This is solid seeded rye, solid seeded beans, no rows. That's probably the way to do it, is like twin rows and do 30s and stuff like that. We don't do any of that stuff. We just go out there with the combine. We run over some of those soybeans. We've got a 30-foot draper head, and we just harvest the rye over the top of the soybeans. And then in the spring, or in the fall, we come back and we harvest the beans. The draper head really helps because when we're harvesting the rye, we only have, 12, 20 inches of material. So the draper head helps because we don't have a lot of material going through the combine when we're harvesting the rye over the top of the beans. Then when we come back and harvest the beans, we have all that soybean and we have all that straw that's still out there. So the draper head really helps to pull all that material through. The reason that we've been doing this is, number one, we want to maximize the value of that cereal rye. Really let that biomass get huge. Let the diversity of the grass and the legume growing together really do its thing. Get that synergistic effect. But also, I want to directly make some money off of that rye. I can harvest that rye as cover crop seed. In my neck of the woods, cover crop seed is for a bushel of cereal rye is 17, 18, 19 bucks a bushel. And it's going, as more and more farmers continue to do cover crops, the price of these inputs isn't going to get any better. So we're like, well, shoot, we're going to keep doing this stuff. We might as well grow our own seed. And we've been using Elbon rye here the last couple of years. I've done VNS. Yankee is a good one. I know guys use a lot, Hazlitt. But some of these different varietals of rye. You can't use a hybrid rye that's on patent and stuff. You can't keep that for seed. But these open pollinated varietals, you can. And keep them and use it. So we've been averaging like 30 bushel rye and then come back and still be able to make 70 bushel beans. It's been amazing. This year, where I had no rain, didn't work so hot. Having all that rye out there and trying to grow a big soybean crop, it hurt us. My rye this year did like 31 bushel, and the beans made like 45. So still good. There's zero fertilizer, zero seed treatment, only one pass of herbicide after we take off the rye. No nothing going out there. And so it's still making really solid money.